So welcome to our in3 podcast. I'm Ninka Kramer. I'm assistant professor at Utrecht University, and I'm very grateful to have taken part of in this EU Marie Curie ITN project these last 3 years, in fact. And I'm a supervisor of one of the 15 early stage researchers, and we tackled the challenge of integrating in vitro assays with in silico or computational tools. And this will also be the topic of this podcast that I will discuss with fellow in3 colleagues. And these our panelists for today are Carolina Nunes, Leonie Franssen, Nicoletta Spino, Susanna Poencha, Pranika Singh, Mark Cronin, Emilio Benfinati, Maxime Culot, and Mariana Novic. Of course, the others are listening on and they can ask questions in the end or have their input spoken. So let me just start. So if I could start with you, Professor Cronin. You're a professor in predictive toxicology at Liverpool John Moores University, and you've led research over the past 20 years actually applying computational tools to predict toxicity, and therefore you're developing alternatives to animal testing. Could you give the listeners just a very short history of what we mean by this ongoing paradigm shift away from animal testing and chemical safety assessment?
Thanks, Ninka. It's really important to put some context behind what we're talking about in this particular podcast. And chemical risk assessment has evolved even in my lifetime. And there's a number of reasons for that. And we could spend the next hour just discussing those. So I'll just summarize some of them that I think are particularly important. And we can start probably almost a century ago, and certainly go into the 1950s with the work of Russell and Birch that set the scene for the 3 Rs. And that gave an impetus, particularly to understand the issues regarding animal testing and doing things in a better and more ethical and less harmful and distressful way. And that's one aspect that drives us on. But more practically as well, we want to do things that are more relevant to humans. At the moment, we're extrapolating up from animal species to humans, and also be able to cope with large number of chemicals, make decisions more rapidly, and so forth. And another enormous driver across the world has been legislation, different sectors of legislation. In Europe, we have the REACH legislation and obviously cosmetics legislation, and they encourage the use of alternatives. And I think in the last decade, we've certainly been pushed by the concept of 21st century toxicology and the opportunities that the new technologies that are just coming online and the new information that we can get very rapidly that's mechanistically based can give us. So there's lots of information we can use and lots of reasons for going to alternatives and wishing to integrate in silico and in vitro.
Yeah, so that's actually really good. You bring up a good point to say. You mentioned this landmark report of the US National Academy of Sciences, toxicity testing in the 21st century, and that this has been, well, you could argue, a major it precipitated a major change in the way we want to conduct toxicity testing, and using what you say, these new techniques, these new technology. I know that it specifically envisions the transition to these in vitro assays with human cells, and these assays are considered central, at least the central part of this new testing paradigm, and arguably not computational or in silico tools, which you work on. You know, if I could ask some of the ESRs here that have been working specifically in these vitro assays. So, for example, Carolina, as an early stage researcher, I know that you've been working on these in vitro assays for the specific purpose. You know, what makes these in vitro assays such, with human cells specifically, such powerful tools?
So I'm part of the brain group, so I'm developing models for brain. And I think the interest here is not just focusing in one organ. It's actually because we have the ability now to derive models for different organs from the same donor. And that is what I think brings strength to our of this new way of testing. And so it's not just about the brain, because you can always find models to answer your questions, and there's a lot already published and that people are working with. The question here is that we can do different models coming from the same donor, and I think that's the strength that we have here.
Yeah. So it's specifically these induced pluripotent stem cells, which allows us to differentiate the same cell from one person to the different organs and really look at it on a multi-organ level. So that's actually a very good point. But then what it sounds like, okay, these are great models. We can make them into different organs. Why is there this necessity then to integrate this with computational tools, or why are these systems not in and of itself sufficient to actually replace animal testing and toxicity testing? Is there someone who could elaborate on that? Susanna Apparenche, I know that you've been integrating these two.
Exactly. Exactly. So I've been working with integration of the kinetics, so basically extrapolating chemical hazard characterization into in vivo. I think, there's probably a lot of reasons, but I see one very clearly. You do in vitro and have results in a specific system. You can always compare to literature, but I think the in silico really helps actually putting your experiments in context of what other people derived and actually predicting something for the future. So it's almost like a way of curating all the data that you get.
Well, that's
and this is what I found. And I think also was one of the things that when I entered toxicology, I thought was really interesting. It's not just like going in literature and just reading papers and trying to see if your experiments make sense, but actually if you have models that already accumulate all this information. So it's an actually smarter way of using your data, let's put it like this, and also more evidence-based, because then actually sometimes we have contrary results, especially in vitro. Sometimes you show a chemical does an effect and sometimes it doesn't. But by curating all these data together, you can actually see what's the most clear result, what's the most probable result of it.
All right. So what you're referring to here, Susanna, is, if am I correct, is that you're collecting the so for example, toxic potencies across toxic potency across a group of chemicals to collate an understanding of its mechanism. With this, there are lots of, I can imagine, so computational or in silico tools in toxicology is quite a broad term. I know that you yourself have been working on physiologically based kinetic models, and I know we just heard from Mark, and he's a guru, I would say, in quantitative structure-activity relationships. And but what you also mentioned, it's also curation, or it means computational also means in bringing together big data into one form, and that would also encompass AOPs, adverse outcome pathways, which might not traditionally be seen as a computational tool. Emilio, you had a point as well.
Yeah, maybe the way to see the in silico method is a very powerful tool to establish a new link between the data and the theory. So, you know, in the past, all the progress of science has been from the data, from the evidence, experience into something which has been formalized into certain theoretical levels. And now the difficulty is that we have many more data. The evidence, the heterogeneity of the data is very complex. So computers can help in this, but also in silico models can be heuristic, so they can generate new knowledge. So this is a new level between the theory and the data. And I think that way we should think about this possibility and exploit it in the best way.
Yeah, that's a good point. So that's coming back to what Susanna then also it's a way to bring together all the individual data points. Max, I know that Max Sklodowska, you've been working on brain barriers for much of your academic career. You had your hand up in response to these in vitro assays and their limitations.
Yes, actually, instead of limitation, it's also that I came to realize and base my thinking on the fact that what we are developing currently, and for example in this project as in vitro models, are already very fancy model. The first ESR we said we use iPSC and we can do donor specific, and that's great, but I don't see the future as such. I don't think that we will, personally, I don't think that the hope for the future is to have a battery of donor that we can test. I rather think that we should try to use those assays to figure out what is going on and identify why is one specific donor responsive to one specific kind or type of chemical. And that's where in my opinion, the in silico should kick in and say, okay, once we figured out a bit of that, now I rely completely on the in silico people to figure out what kind of chemical are going to trigger this, for example, molecular initiating events if we figured it out. And my expectation is that we are going to develop some fancy model to bring some new knowledge, but then I completely rely on the in silico people to take over this knowledge and exploit it, because I don't think it's so realistic to consider a one-by-one replacement, saying we stop with rat and replace them by cells in box. This is not my futuristic view on the future. Maybe I will be retired at the time it will kick in. But my expectation for the future is that the knowledge will be integrated in silico, and then the in silico models will be the realistic way of testing the compounds. And something else that I noticed as well is that we have this tendency of sometimes a bit, in my opinion, underestimating what we already achieved, because we are moving away from animals, and we are well aware of the fact that these animal models are far from being perfect. And I'm pretty sure we somehow already collectively as a scientific field achieve a decent alternative to that. It's just that as scientists we are pushed to go for something much better. So we are not aiming for predicting toxicity in animals, we aim to predict toxicity in humans, and not only one random humans, but we want to be able to do population-based, we want to be able to predict toxicity in different ages and things like that. So we aim for very high expectation.
So that's a very interesting view. I would argue. So you're actually arguing that in vitro is just an in-between step from in vivo to in silico. Am I correct in summarizing that? So in vitro is really just meant to help us get an understanding of the physiology and to use our jargon, adverse outcome pathways and networks, so that we can use our computational tools to actually do the prediction. So that do others agree with that? Or Mariana, you
yeah. Maybe I just wanted to add even before, but I completely agree. And these are complementary tools, actually, this in vitro and in silico. And I think they just fill some gaps, maybe, that are not achieved by one or the other approach. And what I found out is the main difference between this in vitro and in silico tools is actually the in vitro tools produce a lot of information on a smaller system, while in silico tools, especially those that QSARs, for example, they have to have a lot of data and just on a certain amount of field of information, and to use this statistical, let's say, the information that is contained in this large data can be then somehow complemented to the part that you have on a smaller system. And in vitro people have information on smaller systems from the viewpoint of large data that we are using it. And I think from this point of view, these methods are complementary, and I don't think would completely supplement. The in silico is at the end approach to artificial intelligence is like having, but then we have to have so much data and so much information already available that this would be sufficient to decide in future. For this, I think it's not yet so close.
Oh, it's going to be. We'll end on that question, I think, and ask everyone how long it will take. We'll leave that for the end. Mark Cronin, you wanted to respond to Mariana's comment on this notion of the small versus the big in vitro versus the in silico.
Well, not really complement, but maybe extend it a little bit. And I think we just need to remember with particularly QSAR and the way we're using QSAR, it's the endpoint that we need to think about. And we've got different uses. So, for instance, if someone is preparing a dossier for REACH, the purpose of the QSAR there is very specific, and that is to predict the regulatory endpoint, which can be a very, very difficult thing to do for our complex toxicities. We know that. The alternative is, of course, to add to a weight of evidence. So you're predicting something that will add to a weight of evidence that will predict a regulatory endpoint. And the Chemicals Agency, ECHA, are very, very clear on that if you actually read what they're saying and their guidance. I think where we're going now is the future, which we can think about a world where we're going to reconsider, or we've got the opportunity possibly to reconsider chemicals legislation and integrate different types of evidence. So here we will want to use in vitro information to drive the kinetics, the molecular initiating events, and capture that through in silico toxicology. But then what we're having is in silico models for molecular initiating events and kinetics, and somehow we need to turn that into something relating to safety and making a decision relating to safety, which is different from the way, as you said, many years ago I started in QSAR and we were predicting endpoints. And so we're producing a lot of information. And I think, this podcast is really thinking about integration. It is that integration, how we're going to do that, and indeed the need we're going to for it, and what it's going to provide.
That's a very interesting point. So if I could reword or rephrase that, what you're saying is the way we do computational toxicology has changed, probably in light of maybe the in vitro toxicology that has come up. So instead of predicting an endpoint, a broader, like Mariana also mentioned, the lethal dose, for example, you're actually going to use these same types or similar types of models, but here you're predicting actually just a smaller smaller puzzle piece, but then arguably it will be more predictive because it's more mechanistic if you combine that with a suite of in silico and in vitro approaches. Then
I don't think it's changing, and I don't think it's evolving. It's expanding. Our capabilities are expanding. We're still going to want to predict the regulatory endpoints. We're still going to want to predict skin sensitization or whatever it is from structure. We will still continue to do that, and we're evolving and we're getting better methods for doing that. But we're also expanding our approaches, and we can particularly see that in the last few years, going to different endpoints. But what I'm saying, I guess, is let's not lose sight of what we're trying to do and how we're going to use this information.
Yeah, that's a very, very fair point. Emilia Bentonati, I know that you've been working just like with Mark on these QSARs. Has that changed for you too in the way what you're predicting or what you're aiming to predict? Has that expanded, as Mark has said?
Yeah, elaborating, coming back to what has been said by Mark and also Maxime. So let's say on the long term, I must say, he proposed and he claimed that a certain level of in silico will be the only way. I think that this can be achieved only in the hypothesis that at the basis of in silico models we have all the possible pieces of information, which is quite ambitious. So how far are we from this phase where the in silico reaches the truth and the gap of knowledge is very small? I think it depends on the cases. And so the point is that in some situation, for some chemical, for some endpoints, we have sufficient level of reliability, but in many, many other cases, we still need the data. We still need multiple, let's call them line of evidence, multiple data, multiple sources of information. And this, I think, is also what Mark was saying. So we are moving from the approach where we took into consideration the individual pieces. Now we are facing the reality that what we're speaking about is really a mosaic of many, many components. And depending on the level of uncertainty, we need support from the different line of evidence, the different kind of data from omics, in vitro and in silico, and the mechanisms. So I think that this is the process, as Mark was saying, with the idea to cover as much as possible the gap of knowledge.
Yeah, that's a very fair point. I'd like to take it out back, and we'll come back on the issue of where we're going and how long it will take. But just to relate this specifically to in3, so I just want to take the next few minutes. I know that there's a couple of all the users in this panel. You've all been working on specific aspects of this. Of a new next generation risk assessment approach. I know that one of you, Pranika, I know that to point at you now is that you're the one who's been working on this what I would call big data issue. You know, these in vitro assays that your colleagues generated a whole lot of gene expression changes and profiles. What's your views on the necessity of integrating that work for actual risk assessment? What would we need? Like how did your automated pipelines, for example, help in using in vitro data for risk assessment?
So, I mean, my pipeline does not exist if we don't have data. So definitely, like whatever data we get from the in vitro assays, which all my colleagues have generated, and already the existing data, which is already there, especially from the animal models. So integrating them through algorithms and through computational method, I think is very important for the risk assessment, because we can use the data which is already existing and the new one to predict things and to see how our in vitro studies are replicating or are not able to replicate what we already had from the in vivo studies, for example. And this is what I have been also trying to do and see if what we see in the animal data or what we already know from the animal data is something which we can also see in the in vitro data. And for that, like the workflow which I created was to see the gene expression analysis of the gene expression analysis which was induced by the different compounds, and to see if that is something which is already known or is this something which is a new knowledge. So I think both of them kind of goes hand in hand, the in silico and the in vitro or the in vivo, or it's yeah.
Yeah, that's a fair point you're bringing up. So we should not forget we might not want to do any more animal testing in the future, but let's not forget the data that's already there.
Because I think before this, we had only like more experiments were done on animals. So we should not leave it behind. We should keep it with us. Of course, the in vitro experiments now, and they would, I hope, would prove to be an alternative for the animal. But we should not forget, and we should not discard the data which we already have. So that would be the actual integration between the computational methods and the new generation experimental methods, which we are trying to do, especially in our project as well.
Yeah, that's a very fair point. It's like, so we're not only integrating in vitro and computational tools, we're in vitro, in vivo, and computational tools. And that I can imagine these in vivo data. They're essential also for validation purpose, but also for understanding and relating effects across multi-organ systems, for example, but also across chemicals. This, I would imagine, is also the necessary information that would go into adverse outcome pathways, which would be the backbone of how we actually integrate the readouts from different types of toxicity tests, whether it's computational or in vitro and vivo. And so that brings me then to Nicoletta, who's been working on AOPs, and you've been, like Pranika, been collecting data and trying to fashion it into something concrete. Could you explain what you think about how your work is essential in the integration of these new tools for next generation risk assessment?
Well, I think what I'm going to say might be as a way to promote the AOP concept.
Perfect.
But that's the way, indeed, in my case, working with it for the past almost 4 years. And what I saw, or what I've learned actually, was that the amount of evidence that we have needs to be created, needs somehow to be organized, and the AOP allows for us to organize it. And secondly, when we talk about QSR models, for example, we use the data in order to understand patterns in the data. So to understand associations between the molecular descriptors of the compounds of interest. While with AOP, we actually go deeper in understanding the causal effect between the different biological levels that, for example, a chemical can lead to, and finally helping us understand when exactly from an adaptive homeostatic process actually it happens with toxic effect. I would say also that initially the AOP itself was mainly developed based on a systematic review, where it utilized different type of information, including quantitative, including in vitro studies, because you want some sort of quantitative level of evidence to support that qualitative assumption that you make. Nowadays, there is more and more interest in developing new ways of developing AOPs, and using data mining, for example, is one way, but also relying on high throughput data as well to identify new key events, new biological effects. And so the integration is right here because, for example, what Pranika has done can also validate what we know until now, or maybe identify new points further to analyze. Also, using QSR can help to prioritize directions in terms of what compounds maybe need additional investigation. And without in vitro, I don't think things can go further at this point.
Yeah. So like in vitro is still very, very necessary, Max, from what I understand from this discussion. I'll continue this for the last few minutes, and then we go to and end with the last questions of how long will this take. But first, So we've had AOPs. We've looked at this transcriptomics data and the analysis of this big data. But I know there's a computational tool that we've not discussed much yet here, but is I would argue is very central for doing actual and deriving, for example, reference doses or margins of exposure or margins of safety for chemicals, new chemicals, and that's physiologically based kinetic modeling or PBK modeling. I know that Susanna, you've been integrating Carolina's neurotoxicity data from her iPSC systems and trying to predict human exposure scenarios that you would suspect are neurotoxic. Could you just explain what you did to the listener and how that's so vital for next generation risk assessment?
Yes, I can. Thanks, indeed. So we focused mostly actually on the case of the brain with the case study of Carolina and with the specific compound of amiodarone. But I think it's actually very basic in the sense that if we want to use in vitro models to perform chemical hazard characterization. So basically, if we really want to understand these dose response curves or relationships, we actually need this PBK, because one thing is what we test in our in vitro system. So we add a certain dose to the system, and already there, maybe not everything is bioavailable, but I think even more important, like what we add to the system, how is that equivalent to what's in the whole body? So I think these models are actually an essential key, because you cannot really perform risk assessment then, because if all is poisoned depending on the dose, then I think the dose is one of the most important things for toxicology, because I think, and this is still a notion that I think still needs to enter sometimes even in more in toxicology, because we tend to test doses without realizing if they are actually physiological relevant, and you can find anything. It doesn't mean that it actually will happen in your body. And but then there's another point. So the PBK can basically perform the extrapolation of these curved dose responses from in vitro. But we can also, the PBK can be parameterized, and traditional was parameterized maybe with animal data or with clinical data in humans. But this usually, for example, clinical data in humans only available for drugs and drugs that already went through the development phase. But also nowadays, we also have in vitro methods to actually derive these parameters. And that is also where we are working a little bit then. So for metabolism or for oral absorption, understanding how much a compound binds to proteins in serum. So we can also actually perform it nowadays in vitro, and actually also use QSARs already. So I think it's a very interesting development to use both to parameterize the curves, those responses, but also the parameters in the PBK.
Yeah, so you have computational models and in vitro models going into a computational model.
So it's a very good, I think, example of how you actually collate data, very different data from different things to make a whole model, a whole human body, human body, exactly.
Yeah, system to actually a whole risk assessment process. That's well. Thanks for that explanation. I'm going to end the last minute and ask each of you to give the listeners a bit of a time frame that you think we can do without animal testing and toxicity testing, and so that's part 8, and what's the most important next step that we need to get right to make use of next generation risk assessment. Max, Julio, I know that you've been working on this issue for a while. What do you think is the time frame?
Yeah, I don't have a proper answer to that, obviously, but what I'm asking myself is, do we need a ban on the use of animals to start using something else? Because if you look at what happens in cosmetic, maybe the ambition was not as strong as the one of completely stopping animals for all chemicals. And it might be a risky move, because if we don't bring a solution fast, then we might end up with problem. So I don't say this is what should be done, but you should expect that if there is a ban, then obviously we here in this frame of the EU project have benefited already for some funding on that topic, but you should expect a lot more funding and activities from industries. They are already on board and they are already funding our research. But obviously if they have no other possibilities and developing those alternatives to animals, you should maybe expect that this ban would clearly change the game. And then there would be a huge investment for not only industries, but also all countries who want their development to go further to invest in those topics. And with this kind of investment, you should expect some results in the end. And so I don't know whether we are ready to implement that move yet, but in my opinion, this is definitely something that would speed up the process for sure.
Yeah, you can see, you could argue, therefore, that the bans have been pretty effective at getting us to scramble, I would say, for methods to find an alternative to continue putting new chemicals on the market. But when we're not there yet, now there's just no new chemicals coming on the market when it comes to cosmetics. But if you had to put a time frame on it, what if we just look at cosmetics, for example, when can we be doing this and that's accepted? Anybody dare burn their fingers here? Yes, Susanna.
Okay, I'm going to throw in here. So I don't know, for cosmetics, I will say like 10 years, maybe we don't need animal studies. But I think I'm always wondering also what would be the difference between cosmetics and drugs. Of course, cosmetics are not as essential maybe for society, and that's why we put this rule first. But actually from the toxicology point of view or from the kinetics, is it any different? Because if we can do for cosmetics, why can't we do for others, right? And I think that's a better question. So I would put 10 years maybe for cosmetics, just because in my opinion, most of them you actually just put in your skin, so you have a very specific absorption. So they usually, again, from my kinetic bias, would be compounds that maybe don't tend to accumulate greatly in the human body. So usually you can more easily see the effects. Although I would say, not being an expert, that usually I think the immunological points, for example, are some of the most difficult ones to predict. And I'm not really sure if in vitro it's there yet, for example. But also another issue, I think we always talk about ending with animal experimentation. As of course, I think for drugs that are meant to be for human, that makes sense. But we have the whole issue of the pollutants and also, veterinary drugs. And there you will still have animals. And especially for environmental, you still need actually, and even have more diversity maybe of. But even there, then we can ask ourselves, should we also trend to have in vitro models of animals? I think we have ideas of projects, right, of try to actually have in vitro of dolphins or of whatever animals, because actually it's a much more controlled environment in our laboratory to really make sure the whole species or the whole ecosystem is actually healthy. But that's my point of view.
Adds a level of complexity, yeah. What you're saying. That's perfect. Carolina, you had your hand up.
So I think it will be very important how this will evolve for the cosmetics if they will start developing new formulations with new compounds. Because as you said, till now, since the ban, there was not anything really new in the field. They are testing new formulations, but in terms of compounds, everyone is a bit, or from my point of view, a bit afraid of putting something new that they cannot test in animals, at least in Europe. So I think it will be very important when they make that step, and then the others will have courage maybe to follow. But of course, this will depend on how the extrapolations from in vitro to in vivo will also be. And it's a very important part of our project that we are trying to make these connections between in vivo, in vitro, and in silico. So I hope that if these two things grow along and if they are successful, I would say 20 years. So when we, the young investigators, we will arrive to like our 40s, 50s, maybe we will arrive there. I hope I can leave that to the next generation.
Nice. So good goal to go for. Veronica, you had your hand up as a last the speaker. You can wrap it all up here for the listener.
I think I actually agree with Susanna and Carolina both. And I think with the cosmetics, it's easier because it's just the skin. So the route of exposure, it's not going inside our body like a drug does. So, I mean, I'm also not an expert, but I think it will take us like 2 or 3 decades to reach there. And also we need to show and increase the in vitro testing and models and show that they are very robust and we can use them as an alternative to the animal models. Because if we are more convincing on that side, then definitely people would start encouraging and investing for the in vitro testing than on animal testing. So I think we have a long way to go as I see it, because in the end, even as a consumer, I would be a little scared if I have to take a drug which was not tested on a whole animal or something, or just on cells.
Yeah.
So to really show that yes, you can also 100% rely on the in vitro testing, I think we need to put more efforts and more funding and research to show that this is actually an alternative to what we have been doing with animals.
That's a very fair point. That ties up with Max's point earlier. So for cosmetics we have a ban, and it's led to quite, including this research at in3, it's led to quite some new research initiatives. Would bans more, not just in cosmetics, because as Susanna mentioned, tend to be chemicals that should be safe anyways, and applied in a specific route. Would a more general ban increase the funding and the research that we need, or is that a too risky move? I hope our listeners have plenty of information to go by and formulate their own ideas, and do communicate it with us. I enjoyed doing this. Thank you so much for coming up here and letting us know what you think. And up to the next podcast. Thank you.